Welcome to Developmental Biology. Today, we are going to explore one of the most magical phenomena in the natural world, regeneration. Have you ever wondered why a salamander can completely regrow a severed limb, while humans can only heal minor cuts and scrapes? The answer lies in two distinct biological strategies called epimorphosis and morphallaxis. Understanding these processes is not just about studying animals, it is the holy grail of regenerative medicine, holding the key to revolutionary future therapies for human injuries. Let us begin with our first major concept, epimorphosis. Imagine you accidentally break a tall Lego tower that you spent hours building. To fix it, you go to your toy box, grab brand new Lego bricks, and build the missing top part completely from scratch. This is exactly how some incredible animals heal their bodies. Epimorphosis is a highly energetic mode of regeneration, characterized by the active proliferation of new cells to replace lost body parts. The animal creates entirely new tissue to replace exactly what was lost, ultimately restoring the organism to its original size and shape. Now that we understand what epimorphosis is, let us look at how it actually works at the cellular level. When an amputation occurs, the adult cells at the wound site do something extraordinary. They actually forget their current identity. This fascinating process is called de-differentiation. These newly unspecialized stem cell-like cells gather under the wound epidermis to form a mass called a blastema. The blastema cells then rapidly divide and multiply. Eventually, they differentiate back into muscle, bone, and skin to form the new limb. Salamanders and newts use this exact mechanism to flawlessly regrow severed limbs, tails, and even parts of their hearts. This biological process of cell division leads us to a mathematical model. To understand how a blastema grows over time to form a massive new limb from a tiny cluster of cells, we use the standard exponential growth formula. In this equation, n of t represents the total number of blastema cells at a given time t. N0 represents the initial number of de-differentiated cells that formed the blastema right after the injury. The symbol E is Euler's number, a mathematical constant serving as the base of natural logarithms. The variable R represents the intrinsic growth rate of the cells. Physically, when the growth rate R increases, the blastema grows much faster. As time t increases, the total number of cells grows exponentially, providing the raw building blocks for the new limb. Before we move on, let us see this formula in action with a concrete example. Let us say a salamander loses a limb, and a blastema forms with an initial population of 1,000 cells. We are given that the cells divide with a growth rate of 0.1 per day, and we want to find out how many cells will be present after 10 days. We chose the exponential growth formula because it perfectly models active cell proliferation. First, we plug in our known values. We multiply the growth rate of 0.1 by the time of 10 days, which simplifies our exponent to exactly 1. Next, we evaluate e to the power of 1, which is approximately 2.718. Finally, multiplying this by our initial 1,000 cells gives us 2,718. This physically shows how a small pool of cells rapidly multiplies to regenerate a limb in just a few weeks. Now that we have calculated the growth, let us visualize this incredible transformation. On the screen, you're looking at a schematic of a salamander's limb undergoing epimorphosis. On the far left, we see the amputated limb immediately after injury. Following the first arrow to the middle stage, you can clearly see the blastema, that bulbous mass of dedifferentiated cells we just calculated the growth for, gathering right at the wound site. Finally, the arrow on the right leads to the fully regenerated limb, complete with new bones, muscle, and skin. This diagram perfectly illustrates how active cell division builds a complex structure entirely from scratch, restoring the limb to its original size. Having explored how animals build from scratch, this brings us to a completely different survival strategy: morphallaxis. Imagine you have a large clay sculpture of a human, and someone accidentally cuts it in half. Instead of adding new clay, you take the remaining top half and simply reshape the existing clay to form a complete but much smaller human figure. Morphallaxis is a type of regeneration that occurs through the repatterning and remodeling of existing tissues, with little to no new cell division. The organism does not bother making new cells, it brilliantly works with what it already has. Because no new cells are added, the newly regenerated organism is perfectly proportioned, but significantly smaller than the original. To understand how this reshaping happens, we must look at the cellular communication involved. When an organism capable of morphallaxis is cut, the cells that are left behind communicate with each other to figure out their new geographic positions in the damaged body. They then undergo a process called transdifferentiation. Unlike dedifferentiation, transdifferentiation means the cells directly change from one specialized cell type into another, without ever becoming a stem cell first. The most famous example of this is the hydra, a tiny freshwater organism. You can chop a hydra into dozens of pieces, and each piece will remodel into a tiny new hydra. This teaches us about positional information, how chemical gradients tell cells exactly what to become. This concept of positional information relies on a precise chemical coordinate system, which we can model mathematically. To understand how cells know their position during morphallaxis, we use the morphogen gradient formula. In this equation, C of X represents the concentration of a signaling molecule, called a morphogen, at a specific distance X from the head of the organism. C0 represents the maximum concentration at the source. The variable lambda is the decay length, which determines how far the signal travels before it fades away. The negative exponent indicates exponential decay as the signal spreads. Physically, if the distance x increases, the concentration drops. This chemical gradient acts as a map, dictating the ultimate fate of every cell during repatterning. Now that we have our gradient formula, let us apply it to a normal hydra. Suppose a hydra has a morphogen source at its head with an initial concentration of 100 units. The decay length for this morphogen is 2 millimeters. We want to find the concentration at a distance of 4 millimeters down the body axis. We use the exponential decay formula to find exactly what signal the cells at that location are receiving. First, we divide the distance of 4 by the decay length of 2, giving us an exponent of -2. This means we are 2 decay lengths away from the source. Evaluating e to the -2 gives approximately 0.135. Multiplying by 100 gives a final concentration of 13.5 units. Physically, cells sensing this specific concentration will know to become mid-body tissue. This brings us to a critical problem. What happens to this chemical map when the hydra is chopped in half? For morphallaxis to work, the morphogen gradient must scale down to fit the new, smaller body. This is governed by the gradient scaling formula. The formula states that the ratio of the new decay length, lambda 2, to the old decay length, lambda 1, is exactly equal to the ratio of the new body length, L2, to the old body length, L1. By rearranging this equation, we can solve for our unknown variable, lambda 2. Physically, this means that as the total length of the organism decreases, the decay length must also decrease proportionally. This ensures the chemical gradient perfectly fits the new miniature body size, maintaining perfect proportions. Let us see exactly how this scaling works when our 10mm hydra is cut exactly in half. We are given the original length of 10mm and the original decay length of 2mm. Because it is cut in half, our new body length is 5mm. We need to find the new decay length for this smaller piece. We use the rearranged scaling formula because we assume the decay length scales linearly with total body length. We plug in our values, multiplying the old decay length of 2 by the fraction 5/10. 5/10 simplifies to exactly 1/2. Multiplying 2 by 1/2 gives us a final answer of 1mm for the new decay length. Physically, the signal now fades twice as fast, perfectly matching the body that is twice as small. So, we have calculated the new decay length, but why is this mathematical adjustment so important? This chunk of our analysis proves the biological principle of scale invariance. Let us look at the mathematical reasoning. In the original 10mm hydra, the middle is at 5mm. Dividing 5 by the decay length of 2 gives an exponent of -2.5. In our new 5mm hydra, the middle is at 2.5mm. Dividing that by our new decay length of 1 also gives an exponent of -2.5. Both scenarios yield the exact same morphogen concentration. This beautiful mathematical adjustment is the secret behind morphallaxis. It proves that cells are reading their relative position, not their absolute distance from the head, allowing them to create perfectly proportioned miniature organisms. To truly appreciate this scale invariance, let us look at this diagram of hydra morphallaxis. On the left, you see the original, full-sized hydra. Notice the color gradient from red at the head to blue at the foot. This represents our morphogen concentration spreading down the body. In the middle, the hydra is cut in half. Now, look at the right side. The two resulting pieces have not grown in size, but they have reorganized into two fully functional miniature hydras. Most importantly, look at the red to blue gradient on these miniatures. The chemical map has perfectly scaled down to fit their smaller bodies, exactly as our math just proved. This diagram visually reinforces the incredible spatial awareness and flexibility of living cells during transdifferentiation. We have covered a tremendous amount of ground today, so let us summarize our key takeaways. First, we learned that epimorphosis is all about building from scratch. It uses dedifferentiation to form a blastema, which multiplies exponentially to generate entirely new tissue, restoring the original size. Second, we explored morphallaxis, the art of biological remodeling. It relies on transdifferentiation to reshape existing tissues into a smaller, perfectly proportioned miniature without needing new cell division. Finally, we saw how mathematical models, from exponential growth to gradient scaling, perfectly describe these biological miracles. You now have a deep understanding of how chemical gradients and cellular communication allow organisms to survive severe damage. Excellent work today.